The laboratory rat is an excellent specimen for dissection. They are mammals and will therefore have characteristics similar to man. Rats are easily obtained, thus are inexpensive specimens for dissecting, and they have a well-developed reproductive system, leading to a comprehensive study of that system. The rat is of the order Rodentia, which also include the mouse, squirrel, chipmunk, gopher, and beaver. These rodents are the most successful group of mammals, outnumbering all other mammals combined. They have a rapid rate of reproduction, which enable them to occupy new areas and adapt quickly to a changing condition. Rats can be a detriment to man by destroying property and carrying diseases. However, rats are often used in science and cancer research. This is attributed to their similarities to man, plus their rapid reproduction, which allows for several generations within only a few months. Today's specimen has been injected with a colored latex to highlight the circulatory system, the arteries in red, and the veins in blue. As with all dissections, we will begin our study with a review of the external anatomy.
We will begin our study of the rat by examining its external anatomy. Notice that the body is divided into a head, a short broad neck, a trunk, and a tail. The rat is covered with a thick coat of fur. However, the epidermal covering of the tail is quite different. It is covered with reptile-like scales. Look at the head of the rat and notice that it has both a lower lip and an upper lip. The upper lip has a prominent indentation known as the phildrum. We can notice a pair of long yellow teeth known as incisors. Protruding from the lower jaw is a second set of incisors. Enamel covers only the outer surface of the tooth. The soft dentine of the back wears quickly and keeps a sharp edge on the tooth, a characteristic of all rodents. On the dorsal side of the head, we see the external nares, the opening to the ear, which is surrounded by the pinna. The function of the pinna is to channel the sound waves into the ear. The long whiskers that extend out from the side of the head are known as vibrissae. They are sensitive to touch. The eyes are protected by an upper eyelid and a lower eyelid. In the medial corner, we can see the remnant of the nictitating membrane known as the plica semilunaris. It helps in keeping the eye clean. The trunk of the rat is divided into a thorax and an abdomen. Internally, it is divided by a sheet of muscular tissue known as the diaphragm. On the ventral surface of the rat, we can see a row of nipples, which are the external openings to the mammary glands. There are 12 nipples found on the body of the rat. The number varies from mammal to mammal. 3 external openings are found at the caudal end of the trunk. The cranialmost opening is the urethral orifice. It is the opening to the urinary system. The next opening is the opening to the vagina, which is known as the vulva. At the base of the tail is our third opening, which is the anus. It is the opening to the digestive tract. We are now ready to look at the male rat. In the male, we find 2 openings. At the base of the tail is the anal opening. It is covered over by the scrotum, which contains the testes during the breeding season. These sacs are unique to warm-blooded mammals. They allow the testes, which are very sensitive to high temperature, to be held outside the mammal's body. The testes descend only during the breeding season. The ascent and descent of the testes are controlled by the cremaster muscles. The urogenital opening is located at the tip of the penis. It releases sperm and urine from the body. We will now look at the feet of the rat. On the feet we see two specialized derivatives of the skin. We see the claws and the foot pads. This is the vestigial thumb. Rats walk on their toes with their soles elevated. This is called digigrade locomotion. Humans walk on their entire foot, and this is called plantigrade locomotion. And in horses and cattle, this is modified into a hoof, and they are known as ungulates. This concludes the external anatomy. We are now ready to begin the digestive system. To do so, we must make an incision along the mid-ventral line. Using your scissors, cut up the mid-ventral line from the pelvis to the sternum. Now we need to make two transverse cuts. One will be cranial to the lower limbs, the second transverse cut will be made caudal to the upper limbs. It will now be necessary to rinse out the body cavity so that we may see the organs more clearly. After rinsing out the rat, we are ready to study the abdominal cavity. Note the diaphragm, the sheet of muscle that separates the abdominal cavity from the thoracic cavity. In the abdominal cavity, we can see many familiar organs, such as the liver, the stomach, the intestines, kidneys, and reproductive structure. We will examine each of these organs in detail. We will begin with the digestive system. The digestive system begins with the esophagus, which is this tube that connects the stomach to the throat cavity. The esophagus joins the stomach at the cardiac orifice. The first section of the stomach is known as the cardiac. There is a ridge separating the cardiac stomach from the main body of the stomach known as the fundus. The lower portion of the stomach is known as the pylorus. At the terminal end, we see a ring of muscle known as the pyloric sphincter. It controls the movement of food from the stomach into the long coiled intestine. This long coiled intestine is held together by a thin connective tissue known as mesentery. As we look at the mesentery, we can see many small blood vessels running through it. We need to separate the mesentery so that we can see the three parts of the small intestine. The small intestine is quite long and is usually approximately 6 times the length of the rat's body. The first part of the small intestine is known as the duodenum. The next portion is known as the jejunum, and the last portion is the ileum. The ileum joins up with a bag-like structure known as the cecum. The cecum is similar to the vestigial appendix in man, however it is quite large. The cecum has 2 functions, one of which is reabsorption. It reabsorbs water and minerals from the waste material. Another function is it acts as a place of fermentation where the plant matter ingested by the rat can be decomposed. If we would look closely in the area of the last section of the small intestine, we will see some lumpy patches. These lumpy patches are lymph nodes and are known as Peyer's patches. The structure leading from the cecum is known as the large intestine or colon. As it moves towards the cranium, it is known as the ascending colon. As it goes across, it is known as the transverse colon. As it moves towards the tail, it is called the descending colon. These small masses we see in the descending colon are fecal material. The descending colon enlarges caudally to form the rectum. The anal sphincter muscle controls the movement of feces from the rectum to the outside via the anus. There are several other structures associated with the digestive system through which food does not pass. One of these is the liver. The liver is quite a large structure, and technically it has 4 lobes. We see 1, 2, 3, and 4. The rat is unique in that it does not have a gallbladder to temporarily store the product of the liver, which is bile. Bile is a fat emulsifier. The bile will enter the first section of the small intestine, called the duodenum, through a duct called the common bile duct. The liver is also involved in storing glucose in the form of glycogen. Between the stomach and the small intestine is a diffuse gland, which is called the pancreas. The pancreas produces enzymes and hormones. The digestive enzymes produced by the pancreas are secreted into the duodenum. The hormones are secreted into the hepatic portal system, where they are involved in blood glucose regulation. I have removed the skin from the side of the head right below the ear to locate some of the other glands involved. This gland below the ear is called the parotid gland. It produces saliva. The saliva will travel through a duct up to the mouth cavity, where it will moisten the food to begin the digestive process. There are 2 other glands located next to it. One is the sublingual gland. The second is the submaxillary gland. They also produce saliva. In the throat region, we see 4 lymph nodes. These are involved in the immune system of the rat. This long, dark, reddish structure is the spleen. It is part of the circulatory system and functions in the storage and release of blood cells. We need to remove the entire digestive tract. We will remove it from the point where the esophagus enters the stomach down to the colon. Once we remove the digestive system, we will be able to study the reproductive and excretory structures. This ends the digestive system of the rat. The excretory and the reproductive systems are closely linked and are often referred to as the urogenital system. The 2 bean-shaped structures we see here are the kidneys. They are the principal organs of excretion in the rat. They are located in the mid-dorsal section of the abdominal cavity. Usually, the right one is a little bit elevated compared to the left one. The kidneys are embedded in a fatty material. Cranial to the kidney is a small round structure known as the adrenal gland. The kidneys are connected to the circulatory system, from which they remove nitrogenous waste, excess water, salt, and other substances. As we look here, we can see 2 colored vessels entering the kidney. The pink vessel is the renal artery. It enters the kidney from the dorsal aorta. Sitting right next to it is a blue vessel, which is the renal vein. It will leave the kidney and join the caudal vena cava. These two blood vessels are closely attached to one another. Both enter and leave the kidney through the hilus. Also leaving the hilus of the kidney is a white duct known as the ureter. It carries urine manufactured by the kidney to the bladder. We can follow it down to the bladder. In the preserved rat, the bladder appears as a hard, constricted structure. From the bladder, the urine will leave through a short tube known as the urethra. It will join up with the urogenital duct, which will carry the urine to the outside of the body. This bag-like structure is the scrotum. I have cut it open so that we may view the testes and the epididymis located inside. This is the testes. If we look closely, we can see many small, tightly coiled tubes known as seminiferous tubules. They produce sperm. Once the sperm has been manufactured, they are temporarily stored in the epididymis. When the sperm leave the body, they travel from the epididymis down this tube known as the vas deferens. And the vas deferens will go up and around, loop around the bladder, and continue as the urogenital duct. The sperm leave the male genital tract in a fluid suspension. This fluid is called semen. Several glands open into the male genital tract. These glands produce the correct semen conditions for viable sperm. The largest glands are seen here, lateral to the bladder. They are called the seminal vesicles. They are rather rough-looking structures. On the curved inside surface, we see another gland adhering to it, known as the coagulating glands. Below the bladder, we see another pair of glands, known as the prostate glands. The third set of glands are located here. These are Cowper's glands. If we remove the skin from the tip of the penis, we see another set of glands known as the preputial glands. We are now ready to look at the female reproductive structures. In the fatty material just below the kidney are the ovaries. They are small, round structures containing many follicles. Adjacent to the ovary are the fallopian tubes or oviducts. Inside the oviducts are little cilia which beat to draw the egg from the ovary into the uterus. The uterus in the rat is branched to form two uterine horns. The opening from the uterine horn into the vagina is known as the cervix. The egg would follow from the ovary, the oviduct, down the uterine horn, into the vagina, and out through the vaginal orifice known as the vulva. The purpose of the uterine horns is to accommodate multiple births. We have a rat who is pregnant, and we'll now look at her. The masses we see in the uterine horn contain little baby rats. I have opened one of these up to show you its contents. And in here we see this small rat connected by the umbilical cord to the placenta. When it is time for it to be born, this little rat will move down the uterine horn, through the cervix and the vagina, out through the vaginal orifice. The female also has some preputial glands located here on either side of the urinary opening. Also located here is the clitoris. This is similar to the penis in the male. This ends the reproductive structures of the rat. The thoracic cavity has been opened to expose the heart and the lungs. The heart is covered with a gland-like structure known as the thymus gland. It is involved in the immune system of the rat. We need to remove this in order to more clearly see the heart. This is a four-chambered heart which consists of two flap-like structures known as the left and right atria, and two muscular ventricles, the left ventricle and the right ventricle. Leaving the left ventricle is a large pink vessel which curves towards the rat's left and then bends and goes down towards the tail of the rat. This is known as the aorta. It is the major artery in the rat's body. The arch that is formed as it comes out of the left ventricle is known as the aortic arch. And as the aorta passes down the back of the rat, it is known as the dorsal aorta. The aortic arch gives off 5 branches. The first 2 branches are immediate and are difficult to see, especially from this view. They are known as the coronary arteries and supply the heart muscle itself with blood. The next branch is known as the innominate artery. It's quite short and branches immediately into the subclavian, which goes into the shoulder, and the right common carotid artery, which goes up towards the head. This branch is known as the left common carotid artery, which also goes up towards the head. And here we see the left subclavian going into the shoulder. As the dorsal aorta goes towards the tail region, we see many branches branching off of it. One of these branches goes into the ribs and is known as the intercostal artery. Another branch will go into this muscular tissue, which is the diaphragm. That artery is called the phrenic artery. Along the aorta, we see several branches going into the intestines and the mesentery. These are the celiac artery and the cranial mesenteric artery. This artery is going to the adrenal gland and is known as the adrenal artery. There's also a renal artery going into the kidneys. Further down, we see a large branch coming off and going into the legs. This is known as the common iliac artery. If we look carefully, we see a small one going into the tail region, which is called the caudal artery. Also seen in the rat are several veins. Veins are vessels that carry blood back to the heart. They have thinner walls than arteries. They also have valves which prevent the backward flow of blood. They are stained blue in the rat. The large blue vessel that can be seen arching out of the right ventricle is the pulmonary arch. It divides into a right and left pulmonary artery. These major arteries carry blood from the heart to the lungs. The pulmonary arteries carry deoxygenated blood, and while the blood is in the lungs, it will be oxygenated and returned to the heart through the pulmonary veins. 2 major veins return blood from the body of the rat to the heart. They are the cranial vena cava and the caudal vena cava. The rat is unique in the fact that it has a right and a left cranial vena cava. The left vena cava curves around the underside of the heart and joins up with the right vena cava at the sinus venosus. There are 5 major veins that empty into the cranial vena cava. These veins return blood from the ribs and from the head region of the body. There are also major veins along the trunk of the body which dump into the caudal vena cava. They lay adjacent to the arteries. One vein we see here is the common iliac vein. This is the spermatic vein, the renal vein, adrenal vein. All of these veins dump into the caudal vena cava, which will carry the blood up to the right atrium of the heart. From the atrium of the heart, the blood will pass into the right ventricle, out through the pulmonary arch to the lungs, where it is oxygenated, it will return through the pulmonary veins to the left atrium, into the left ventricle, and out through the aortic arch to the rest of the body. This completes the circulatory system of the rat. Air enters the respiratory tract through the external nares. It'll pass through the glottis into this wide-shaped structure we see here, which is known as the larynx. The larynx is otherwise known as the vocal box. Lateral to the larynx are red-shaped structures known as the thyroid. Small parathyroid glands are embedded in the thyroid. Continuing from the larynx is a long tube known as the trachea. As you look at the trachea, you can see small rings going across it. These are made out of cartilage and prevent the trachea from collapsing. If we follow the trachea down, we will see that it will branch near the heart into a right and left bronchus. The bronchus will lead into the lung, where it will branch again into bronchioles. It will continue to branch and rebranch until it gets into its final division known as alveoli. It is here where the gas exchange occurs. The rat has a 5-lobed lung. It has 1 left lobe and 4 right lobes. We can see blue vessels entering the lungs. These are the pulmonary arteries. If we look closely, we can also see pink vessels leaving the lungs. These are branches of the pulmonary vein. This completes the respiratory system of the rat. The cranium must be opened to expose the brain. This requires a great deal of time and care. We will examine one that has already been opened. To really study the brain structures, we need to take this brain out of the cranial cavity and examine it. I have one here for us to look at. This is the olfactory lobe. Caudal to it is the cerebrum. It is divided by a deep cerebral fissure. It divides the cerebrum into a left hemisphere and a right hemisphere. Here we have the penile body. This is the cerebellum. It is a highly convoluted structure. It consists of 2 lateral hemispheres and a medial vermis. The deep fissures are known as sulci, and the folds are gyri. Off to the sides, we can see here the paraplococcus. The anterior portion of the brain is known as the medulla oblongata. It is divided by a deep groove called the sulcus medianus. The spinal cord will continue posteriorly down the vertebral column of the rat. Paired spinal nerves will leave the spinal cord at intervals. On the ventral surface of the brain, we again see the spinal cord, the medulla oblongata, the paraplococcus of the cerebellum. This is the trigeminal nerve. Here is the optic chiasm and the optic nerves, olfactory lobes. Located in this area would be a reddish gland that hangs down from the brain, which is the pituitary gland. This concludes the nervous system of the rat.
The dissection and anatomy of the rat has shown the similarities between this primitive mammal and man. The laboratory rat proves to be an asset to the science field, allowing for many different types of research and also for mammalian dissections, as you have